Hello, Public Health 4040 students. This is Fundamentals of Epidemiology, and I'll be covering material that's found in Chapter 10, where we're going to be talking about data interpretation issues. Think about this, the way the series of this class has been going, where we've talked about where data comes from, what we do with the data, or what kind of questions we want to be able to ask. And then we've talked about study designs that are out there, how to analyze the data, and that's what we talked about in Chapter 9. As well as in earlier chapters when we talked about odds ratios and relative risk. And then the real question is that now that we have this data, we have these findings, we need to be able to interpret these findings, and that's what we're going to talk about in this particular chapter. First thing that's really important is that it's always important to talk about the validity of a study design. The question that always comes up is that did we actually obtain the data and the findings that we intended to find with the data that we collected or the study that we did. You have to evaluate that particular study design and the methodology that was used within a particular study. There's numerous examples within the media where individuals, media sources will take particular specific findings and extrapolate them out to a general public when it's not really all that appropriate. Or they will make really significant inferences based on certain types of findings when you may have a very small sample size, or where you may just not collect the initial exposure does not really match the particular outcome. So you have to always be looking when you're reading literature, when you're reading in your textbook, how valid are these findings? How valid is this particular study? And then you have to ask yourself, does this study actually, is it usable, or can we extrapolate to other study populations that are out there? So a couple of things that you need to think about when we talk about validity is we'll break it apart into based on internal validity and external validity. Internal validity is where a study is said to have internal validity when there has been proper selection of study groups and a lack of error in measurement. Now, in a perfect study, that would actually be great. And that's the ultimate goal, is to be able to actually link an observed effect to a particular exposure. And if you think about the different types of study designs that are out there, community trials as well as intervention studies or clinical trials, those have the highest level of internal validity. But it's not always feasible to be able to do a clinical trial. And so you have to look at, like, for example, case control studies or cohort studies. And when we think about the data that was collected, are we actually being able to link that observed effect to the exposure? And a couple of things that you would look at when you're looking at internal validity is, and you need to be concerned with, is what type of measurement was completed specific to an exposure. What kind of measurement was done when we look at the outcome? And are we able to actually make an association between that exposure and the disease? So we'll break it apart when we talk about those appropriate measurements, specifically looking at exposure. And a couple of things that you need, a couple of really good questions that I listed here that helps you think about exposure and whether there was an appropriate measurement made. For example, were the study instruments validated or study samples standardized? And when I talk about a study instrument validated, you ask yourself, wait a second, how do I relate to that if I'm looking at, for example, a vaccine trial where we know that we have a particular vaccine or maybe a new vaccine that's about ready to go out onto the market, and then we inject it into a particular study population, and then we can collect antibody samples or blood samples and determine whether or not that particular study instrument is valid. And so you sit there and you question how it doesn't really make sense. And the way that I like to relate this to is that is where there's a number of epidemiological studies where they use surveys or survey instruments, and you have to always look at these survey instruments and really ask yourself, was this particular survey vetted, and did it actually measure is it actually going to measure what we intended to measure? And are we going to be able to interpret the data that comes from that? And then you also have to ask yourself specifically, were the study samples standardized? As in, are we able to actually take those samples that we collected, those survey instruments, and be able to use them in a way that we can interpret the data effectively? And then you have to ask yourself, when you look at exposure, is that are we actually measuring what we intend to measure? All too often we collect data, or scientists will collect data, and I'm not talking so much about it from a laboratory standpoint, but when we're working in a public health scenario where we're actually working with people, are we actually measuring what we intend to measure? And there's numerous examples. For example, you could look at it within healthy eating habits within an elementary school, and you say, well, that's not hard. We're going to go and look at the types of foods that are being served, and then do we see those behaviors that are associated with that? But you have to ask yourself, what is the data that we're actually going to collect, and are we actually appropriately measuring and obtaining the outcomes that we're expecting to see? And that leads me to outcomes, as in, did we actually identify all the eligible subjects and locate them, and were they included within our study? And were all the endpoints actually identified, or did we actually lose any to follow up? So, for example, with this, where it's an issue, is that there's numerous infectious diseases like Zika virus or West Nile virus, in the sense that you have to look at the types of study designs that are out there. We may lose a number of individuals to follow up, or we may lose where we may not actually identify all the eligible subjects that could be a part of the study. As I said, with West Nile virus or Zika, there's numerous individuals that become infected that never even know that they're infected. And so here we have lost individuals, as well as data that comes with that, and we will then potentially, from an internal validity standpoint, maybe miss some of those findings. And then, is there an actual association between the exposure and the disease? And did we actually properly analyze it? And did we make the appropriate adjustments that may influence those results? Or did we make adjustments to those findings that may influence it in an inappropriate way? And then you have to ask yourself, is that did we do all the necessary analyses in order to be able to critically analyze? Or did we omit certain analyses, or did scientists do that because the study findings didn't go their way and they still wanted to be able to present that data? All right, moving on from internal validity, we'll talk a little bit about external validity, and this is actually where we're going to be able to take the data from a smaller study, and we're trying to actually understand whether or not these findings from this smaller study would actually be generalizable to a larger population. So external validity actually implies the ability to generalize beyond a set of observations to some universal statement. And as I said, as I'm kind of leading to, it's more than extrapolating from a sample population to a target population. And you have to ask yourself, how so? You have to be able to have a study design that's not only internally valid, but that is externally valid, to where you can use this data as well as these findings and be able to make general statements on this larger population. As I talked already in this course before, when I was at CDC, I worked in a surveillance group called the Active Bacterial Core Surveillance Project, and this is where we actually had a study where we looked at meningitis rates in 10 different study sites throughout the throughout the entire part of the United States. Different states were involved in this particular study, and the reason why we were able to actually determine whether or not this particular study design was externally valid was because of the way the study was actually set up, and the particular study populations were representative of what we would expect to see as we moved it to this larger target population. We also did analyses within this particular study that wasn't just statistical or mechanical, and that we were able to actually determine that these findings are generalizable to the U.S. population. It's also important when we talk about external validity is you have to understand the relevance of these particular findings, and can we take these findings from this smaller study and be able to go beyond or generalize it to this larger target population. And that's a real issue when we talk about it from a clinical trial standpoint, is because we'll talk about it later on in this particular discussion, but clinical trials have set controls as to what type of individual can participate within this clinical trial. And it's not just whether or not they had cancer or not. For example, a lot of clinical trials, when you're looking at new cancer drugs, they have to be at a specific stage. And so it may not be externally valid with certain types of study designs or outcomes when you try and extrapolate it to all stages of cancer. So as we move on and talk about in depth about internal validity and external validity, there's things that you have to ask yourself as to what can make a study design and what can make the outcomes that we're actually trying to interpret make them invalid. There's sources of error within many different types of epidemiologic research that you need to take into account. And some of those errors that we're going to talk about are random errors, and they reflect more the fluctuations around a true value or perimeter because of the sampling variability. There's different factors that contribute to random error. And then what's even of more concern is systematic errors, or where we have measurement bias, and we'll talk about each of these in quite a bit of detail.